Good morning, everyone. This is Ainsworth, and today I want to walk you through one of the most fascinating nutritional experiments that is quietly gaining serious attention among metabolic researchers, longevity scientists, and clinicians who study how the human body responds to extreme dietary simplification. I am talking about eating nothing but sardines for 3 full days. Now, before you dismiss that as internet nonsense, I need you to understand something. What happens inside your body when you remove every food source except one of the most nutrient-dense organisms on the planet is not trivial. It is a controlled metabolic event that activates a cascade of hormonal, cellular, and neurological changes that most people have never experienced because they have never given their body the specific conditions required for those changes to occur. And today I am going to walk you through exactly what happens, hour by hour, day by day, what your insulin is doing, what your liver is doing, what your brain is experiencing, what is happening at the cellular level that you cannot feel, but that is fundamentally altering the quality of every tissue in your body. And I am going to explain why the sardine of all foods is almost perfectly engineered by nature to create these conditions in a way that pure water fasting cannot replicate and that most other foods would actively prevent. But before we begin, if you are new to my channel, hit subscribe right now and turn on the bell icon. Because this is what I do. I take the real science, the actual mechanisms, the physiology that matters, and I deliver it to you in a way that allows you to make informed decisions rather than relying on the trends. Now let me begin with the question that matters most. Why sardines? Why not chicken breast? Why not eggs? Why not simply fasting on water alone? Because the answer to that question is what makes this entire protocol work, and it is the part that almost every surface-level explanation of sardine fasting gets incomplete or entirely wrong. The sardine is not chosen because it is convenient, or because someone on the internet decided it was trendy. The sardine is chosen because it occupies a position in the nutritional landscape that almost no other single food occupies simultaneously. It is extraordinarily nutrient-dense. It is extremely low in carbohydrates, effectively zero. It contains high-quality complete protein in a moderate amount. It delivers one of the highest concentrations of omega-3 fatty acids of any food that exists. And because you consume the entire animal, including the soft, edible bones, it provides meaningful amounts of calcium, vitamin D, selenium, coenzyme Q10, taurine, and vitamin B12, at levels that exceed your daily requirement in a single serving. Now here is why that combination matters metabolically. When you eat only sardines for 3 days, you are creating a very specific internal environment. You are removing carbohydrates entirely. That means blood glucose is not being replenished from dietary sources. Insulin, which rises in response to incoming glucose and to a lesser extent incoming protein, drops dramatically and stays low. And when insulin drops and stays low for an extended period, a sequence of metabolic events begins that is remarkably similar to what happens during a pure water fast, but with one critical difference. Your cells are still receiving essential nutrition. Your muscles are still receiving amino acids. Your brain is still receiving the raw materials it needs for neurotransmitter production and membrane repair. Your mitochondria are still receiving the coenzyme Q10 and the omega-3 fatty acids that support their function. You are fasting and nourished at the same time. That is the entire principle. And that principle is what separates this from a standard fast, from a crash diet, and from the kind of extreme calorie restriction that breaks the body down rather than building it back stronger. This concept has serious scientific roots. Dr. Walter Longo's research on fasting mimicking diets demonstrated that when you keep calories low, keep carbohydrates minimal, and modulate protein to a moderate level, the body enters a metabolic state that mirrors many of the most beneficial aspects of a complete fast. Insulin drops. The IGF-1, an insulin-like growth factor 1, declines. AMPK, the cellular energy sensor, activates, and autophagy, the internal cellular cleanup system that clears damaged proteins, dysfunctional mitochondria, and accumulated cellular debris, switches on. All of this while the body is still receiving nutrition, not abundant nutrition, not growth signaling nutrition, but maintenance nutrition. survival-grade fuel that keeps the essential systems running while allowing the deep repair systems to activate. Now, I want to be precise here because precision matters. A sardine-only protocol is not a textbook fasting-mimicking diet in the way Dr. Longo defined it. The protein content in sardines is higher than what a strict fasting-mimicking protocol specifies, and that additional protein does create some degree of mTOR activation that a lower protein approach would avoid. But the practical metabolic outcome is remarkably close. Insulin is profoundly suppressed. Ketone production begins rapidly because there are no carbohydrates to prevent it, and the caloric intake, typically somewhere around 600 to 800 calories per day, depending on how many tins you consume, is low enough that the body shifts into an unmistakable conservation and repair state within the first 24 hours. If you are still with me, comment below the word yes. I want to know how many of you are genuinely paying attention, because what I am about to walk you through next is the hour-by-hour breakdown of what actually happens inside your body across 3 days of this protocol. And understanding this timeline is what transforms sardine fasting from an odd-sounding idea into a tool you can use with confidence and intention. Also, if you're enjoying the value so far, make sure you give me a thumbs up on this video and subscribe to my YouTube channel for more science educational lectures. Now, let us begin with day 1. And I want to start with the morning because how day 1 begins sets the hormonal trajectory for everything that follows. You wake up. Instead of your normal breakfast, you open a tin of sardines. Now, psychologically, this is the hardest moment of the entire 3 days for most people. Not because the body is struggling. The body is fine. It is the mind that resists. Eating fish at 7 in the morning feels wrong because it violates every conditioned expectation about what breakfast is supposed to look like. But the body does not care about your expectations. The body cares about what it is receiving at the molecular level, and what it is receiving is approximately 20 grams of complete protein, a substantial dose of omega-3 fatty acids, virtually zero carbohydrates, and a mineral profile that most breakfast options cannot come close to matching. In the first several hours of day 1, you will likely feel entirely normal. Your body is still running primarily on glycogen, the stored glucose in your liver and muscles from the previous day's meals. Blood sugar is stable. Insulin is present, but beginning to decline because no carbohydrates are arriving to stimulate its release. The protein in the sardines does produce a mild insulin response, but it is modest compared to what a carbohydrate-containing meal would generate. And critically, it is accompanied by a simultaneous glucagon response. Glucagon is the counter-regulatory hormone to insulin. When protein arrives without carbohydrates, both insulin and glucagon rise together. And the net metabolic effect is very different from the insulin dominance that carbohydrate-containing meals produce. By the afternoon of day 1, something interesting begins. You expect to be hungry. Most people anticipate that eating 600 calories of sardines for an entire day will produce the kind of gnawing, desperate hunger that makes concentration impossible and willpower irrelevant. But that is not what happens. The hunger is present but manageable, and the reason is that sardines are activating satiety through multiple independent pathways simultaneously. The protein is stimulating peptide YY and cholecystokinin, 2 gut hormones that signal fullness to the brain. The omega-3 fatty acids are modulating ghrelin, the hunger hormone, reducing its amplitude. The fat content is slowing gastric emptying, extending the period of satiety after each serving. And there is emerging evidence that the calcium content from the edible bones contributes to nutrient-specific satiety signaling, a mechanism by which the body reduces hunger when it detects that essential mineral needs are being met, regardless of total caloric intake. So you are eating far fewer calories than normal, but the hunger signal is being managed from multiple directions at once. This is not willpower. This is biochemistry working in your favor because the food you chose happens to hit every satiety pathway simultaneously. Have you tried the 3-day sardine diet? Did you notice bad breath or a lingering fishy smell? Share your experience in the comments below. And don't worry if that happened to you. It's actually more common than most people realize, and there's a simple reason behind it. Now, by the evening of day 1, glycogen stores are running low, the liver has been releasing stored glucose throughout the day to maintain blood sugar for the brain, but the supply is depleting because no dietary carbohydrates have arrived to replenish it. Insulin has fallen to levels meaningfully below what your body typically maintains during a normal eating day, and glucagon has risen in proportion. This rising glucagon is the signal that tells the liver to begin mobilizing stored fat. Fatty acids begin entering the bloodstream from adipose tissue. The liver begins converting some of those fatty acids into ketone bodies. Beta-hydroxybutyrate, the primary ketone, is starting to appear in measurable amounts. You may notice a slight energy dip in the late evening of day 1. This is the transition period. The body is switching fuel systems. The main power grid, dietary glucose, has gone offline. The backup system, fat oxidation and ketone production, is spinning up but has not yet reached full operating capacity. This transition dip is temporary, predictable, and not dangerous. It is the metabolic equivalent of the lights flickering for a moment when the generator kicks on. And conveniently, it tends to coincide with the time you are going to sleep anyway. If you're enjoying the value in my lecture, please make sure you hit that red subscribe button and turn on the bell icon so you never miss any of my future lectures. Also, give me a quick thumbs up on this video so I upload more science lectures on this channel soon. Now, day 1 ends with you going to sleep in a state that is hormonally and metabolically extraordinary compared to a typical night. Insulin is low, glucagon is elevated. Growth hormone, which rises naturally during sleep and rises further when insulin is suppressed, is going to reach levels tonight that it does not reach on a normal eating day. And that elevated growth hormone is going to do 2 things simultaneously while you sleep. It is going to protect your lean muscle tissue with a powerful preservation signal, and it is going to accelerate the mobilization of stored fat for fuel. Your body is not breaking down muscle overnight. It is explicitly protecting muscle while burning fat. These 2 processes are not in competition. They are operating together because the hormonal environment you created by eating only sardines today made both of them possible simultaneously. And overnight, autophagy, the cellular cleanup system, is beginning its first meaningful activation. Insulin has been low for long enough. AMPK, the energy sensor, has been activated by the caloric restriction and the absence of carbohydrates. mTOR, the growth and building signal, has been suppressed because the overall nutrient load has been so much lower than what the body normally receives. The conditions that autophagy requires to switch on are assembling. The cleanup crew is arriving. The preliminary work is beginning. Damaged proteins are being tagged. Dysfunctional mitochondria are being identified, and you are asleep through all of it. The most powerful cellular work of day 1 is happening while you are unconscious, and it is happening because the hormonal environment you created through a single day of sardine-only eating gave your body permission to begin the repair work that constant eating never allows. If you are finding this lecture valuable, subscribe to my channel right now and turn on the bell icon because we are only through day 1, and the physiology of day 2 and day 3 is where this protocol becomes genuinely extraordinary. Give me a thumbs up on this video and share it with someone who has been curious about sardine fasting but has never understood the actual science behind why it works. Now let us move into day 2, and I want you to pay close attention here because day 2 is where the sardine fast stops feeling like a dietary experiment and starts feeling like something your body was waiting for permission to do. You wake up on the morning of day 2 and something is different. Not dramatically different, not the kind of difference you have to talk yourself into noticing. It is a quiet, unmistakable shift in how your brain feels. There is a clarity to your thinking that was not present yesterday morning. The mental fog that you may not have even realized you were carrying, the low-grade noise that constant blood sugar fluctuation creates in the background of every waking hour is reduced. And the reason is measurable. Overnight, your liver completed the metabolic transition that began during the evening of day 1. Glycogen stores are now functionally depleted. The liver has shifted its primary activity from glycogen breakdown to fatty acid oxidation and ketone production. Beta-hydroxybutyrate levels have risen meaningfully. Your brain, which was running almost entirely on glucose 24 hours ago, is now receiving a significant and growing proportion of its fuel from ketones. And ketones are not an inferior backup fuel for the brain. They are in several specific ways a superior one. Ketones produce less oxidative stress during cellular combustion than glucose. They stabilize neuronal membrane potentials in a way that reduces the excitatory signaling that creates mental noise, distractibility, and the subtle anxiety that most people experience as a baseline state without recognizing its metabolic origin. The brain running on ketones is a brain operating in a quieter, more stable neurochemical environment, and the subjective experience of that, the calm focus, the sense that thinking requires less effort, the reduction in mental chatter that has nothing to do with your circumstances and everything to do with your fuel source, this is what people consistently describe on the morning of day 2. Not because they are imagining it, because the neurochemistry has genuinely changed. Now, underneath that subjective clarity, your body is doing extraordinary metabolic work. Insulin has been suppressed for over 24 hours, and that sustained insulin suppression has created consequences that go far beyond simple fat burning. Your liver is pulling from its own stored fat. Hepatic fat, the fat that accumulates inside liver cells and that drives insulin resistance, inflammatory signaling, and metabolic dysfunction, is being mobilized as fuel. For anyone carrying excess liver fat, and studies suggest that roughly 25 to 30% of the general population does, this is among the most therapeutically significant metabolic events that can occur outside of specific pharmaceutical intervention. The liver is cleaning itself. Not because you took a supplement that claims to support liver health, because you created the hormonal conditions, sustained low insulin and elevated glucagon that allow the liver to access and burn its own stored fat. Visceral fat, the deep abdominal fat that wraps around the organs and that produces inflammatory cytokines linked to cardiovascular disease, type 2 diabetes, and metabolic syndrome, is being accessed preferentially during this window. Visceral fat has a rich blood supply and a high density of receptors for the hormones that are currently elevated, particularly glucagon and catecholamines. The hormonal environment of day 2 makes visceral fat metabolically exposed in a way that subcutaneous fat is not yet. Your body is targeting its most dangerous fat stores first, not because it is making a conscious choice, because the biology is designed to prioritize the removal of metabolically active, hormonally disruptive visceral fat when the conditions allow it. And the conditions you have created through 30 hours of sardine-only eating are precisely the conditions that allow it. Autophagy at this stage is no longer preliminary. The cleanup crew is not running preliminary checks, it is actively working. Damaged proteins that were contributing to cellular inflammation are being broken down and their amino acid components recycled into raw materials for building newer, more functional replacements. The dysfunctional mitochondria, the ones that were producing energy inefficiently while generating excess reactive oxygen species, are being identified and tagged for removal through the specialized autophagy process called metaphagy. And here is what makes the sardine protocol distinct from a water fast at this same time point. During a water fast at 36 hours, autophagy is active, but the body is receiving zero nutritional support. The cleanup is happening, but the raw materials for rebuilding are not arriving. During a sardine fast at 36 hours, autophagy is active and the body is simultaneously receiving omega-3 fatty acids that are being incorporated into cellular membranes, coenzyme Q10 that is supporting mitochondrial electron transport chains, selenium that is powering glutathione peroxidase and the antioxidant defense system, and amino acids that are available for the construction of new cellular components as the old ones are cleared. The cleanup crew and the construction materials are present at the same time. This is the fundamental advantage of a nourished fast over a pure fast. The demolition and the rebuilding are not sequential, they are simultaneous. Comment below and tell me whether you have ever experienced the mental clarity shift that happens during ketosis, whether it was through sardine fasting, extended fasting, or a ketogenic diet. I want to know how many of you have felt this and whether it changed how you think about the relationship between food and cognitive performance. Now let me walk you through what happens with hunger on day 2 because this surprises almost everyone. You expect hunger to be worse on day 2 than day 1. It is not. For most people, hunger on day 2 is less than day 1, significantly less, and the mechanism is straightforward. Ketones directly suppress ghrelin. The longer you remain in sustained ketosis, the more effectively ketones blunt the hunger hormone. The ghrelin waves that arrive predictably on day 1, timed to your habitual meal schedule, become quieter on day 2. They arrive with less amplitude, they pass more quickly, and between the waves there is a genuine absence of hunger that feels different from simply being distracted from hunger. It feels like the signal has been turned down at the source. This is compounded by the ongoing satiety signaling from the sardines themselves. Each serving you eat continues to deliver the protein, fat, and mineral profile that activates every major satiety pathway. You are eating roughly 600 to 800 calories per day, and you are not suffering. You are not white-knuckling through cravings. The biochemistry is managing the hunger for you because the food you chose and the metabolic state you created are working together to make low-caloric intake feel sustainable rather than agonizing. Now, here is something else happening on day 2 that deserves its own discussion. The omega-3 fatty acids from the sardines are not just sitting passively in your bloodstream waiting to be burned for energy. They are being actively incorporated into cellular membranes throughout the body. EPA and DHA, the two primary omega-3 fatty acids in sardines, are structural components of every cell membrane in your body, with particular concentration in brain tissue, retinal tissue, and cardiac tissue. During a period when autophagy is clearing damaged cellular components and the body is in active repair mode, having a continuous supply of these essential fatty acids means the membranes being rebuilt are being constructed with optimal raw materials rather than whatever happened to be available. This is a nuance that pure water fasting cannot replicate. The repair is happening, but the quality of the repair depends on what building materials are available, and sardines happen to provide building materials that are specifically relevant to the structures being rebuilt during cellular renovation. It is as if the demolition crew brought premium construction materials with them when they arrived for the job. Also, if you are finding this lecture giving you a completely new and more precise understanding of how sardine fasting works at the biological level, share it with someone who has been curious about trying it. Now let us arrive at day 3, and I want you to understand that day 3 is not simply more of day 2. Day 3 is where the protocol reaches its deepest physiological expression. The metabolic state is fully established, fully stable, and fully committed. Ketone levels are at their highest point of the entire 3-day period. The brain is receiving the majority of its fuel from beta-hydroxybutyrate, with gluconeogenesis from the liver supplying the small remaining glucose requirement for the brain regions that still depend on it. The mental experience of day 3 is what converts skeptics. It is not the gentle clearing of day 2 amplified, it is something qualitatively different. A settled stillness of cognitive function that people consistently describe as unlike anything in their normal experience. The neuroinflammation that chronic blood sugar fluctuation produces has been absent for nearly 3 days. BDNF, brain-derived neurotrophic factor, which supports neuronal repair and synaptic connectivity, is elevated. The glymphatic system has been operating in a reduced metabolic noise environment for multiple sleep cycles. The brain at this stage is not just running on cleaner fuel, it is running in a cleaner environment. Autophagy on day 3 is at its most sustained and significant level of the protocol. And here is where the sardine fast creates something that shorter protocols do not achieve at the same magnitude. The combination of profoundly suppressed insulin, declining IGF-1, sustained AMPK activation, and deep mTOR suppression has been maintained for long enough that the cellular repair work is no longer incremental. It is systemic. Cells throughout the body are clearing accumulated damage that has been building for weeks or months of normal eating. The immune system is undergoing its own version of this cleanup. Older, less efficient immune cells that have been contributing to chronic low-grade inflammatory signaling are being cleared. The immune system is being pruned, not suppressed, refined. And the sardines continue providing the nutritional foundation that makes this deep repair work possible without the depletion risks of a pure water fast. The selenium is supporting antioxidant defense while autophagy generates free radicals as a byproduct of cellular breakdown. The B12 is supporting methylation pathways that are essential for DNA repair and gene expression regulation. The taurine is supporting bile acid production and cellular osmotic balance. The protein, while moderate, is sufficient to prevent the kind of muscle catabolism that extended water fasting can begin to produce in individuals with lower body fat reserves. Growth hormone on day 3 is at one of its highest sustained levels of the entire protocol, and its message to every tissue is unambiguous. Protect lean mass. Burn stored fat. Repair damaged tissue. This is not the body in crisis. This is the body in a state of intelligent, resource-efficient renovation that human biology evolved to perform during periods when food was scarce, but survival was not threatened. Tell me in the comments if you have ever completed 3 full days of any restricted eating protocol, whether it was sardine fasting, water fasting, or any form of calorie restriction. I want to know what your experience was and whether the science I am describing today matches what you felt. Now let me address something that is critically important. How you end this protocol matters as much as how you execute it, because breaking a sardine fast incorrectly is one of the most common ways people undo a significant portion of the cellular work they invested 3 days achieving. After 3 days of profoundly low insulin, your body is in a state of maximum insulin sensitivity. Your pancreas, which has been producing minimal insulin for 72 hours, is primed to respond with a vigorous insulin release to the first significant carbohydrate-load it encounters. And if that first meal after the sardine fast is a large plate of refined carbohydrates, the resulting insulin spike in the context of maximum insulin sensitivity will be amplified dramatically compared to the same meal eaten on a normal day. That amplified spike does not just end the repair phase, it creates a fat storage signal of greater magnitude than normal eating would produce. You will be actively working against everything the 3 days accomplished. The correct refeeding approach follows a deliberate sequence. First, shift the macronutrient ratio. You have been eating moderate protein and moderate fat for 3 days. The first meal after the sardine fast should emphasize protein while reducing fat. A clean whey protein shake, scrambled eggs, a small portion of lean chicken breast. The increased protein relative to fat produces a controlled insulin response that signals the body to shift from repair mode into rebuilding mode. This is the transition your body needs. The fast performed the cleanup. The refeed provides the construction signal and the building materials for cellular reconstruction. Amino acids from that first higher protein meal are directed toward the tissues that autophagy has been preparing for renewal. The mTOR activation from protein, which you suppressed for 3 days to allow autophagy to run, is now exactly what you want. mTOR switches on. The rebuilding begins. After the initial protein-forward meal, gradually reintroduce normal foods over the following 12 to 24 hours. Whole food sources, vegetables for fiber and micronutrients, healthy fats in moderate amounts, and carbohydrates reintroduced slowly and from whole food sources rather than refined or processed options. The gradual reintroduction allows the digestive system, which has been operating at reduced capacity for 3 days, to resume normal function without the distress that a sudden large mixed meal would produce. Now, I also must be completely honest about who should not attempt this protocol. If you are pregnant or breastfeeding, this is not appropriate. If you have a history of disordered eating, any form of restrictive eating protocol carries risks that require professional guidance. If you are on medications for diabetes, particularly insulin or sulfonylureas, the sustained low insulin state could produce dangerous hypoglycemia without medical supervision. If you have significant kidney disease or cardiac arrhythmias, the metabolic demands and electrolyte shifts require clinical monitoring. And if you are significantly underweight, restricting calories to this degree adds physiological burden to a system that needs nourishment, not further restriction. For metabolically healthy, well-nourished adults who approach this with adequate preparation, proper hydration, and a thoughtful refeeding plan, a 3-day sardine protocol is one of the most elegant metabolic reset tools available. It delivers many of the deep cellular benefits of extended fasting while maintaining the nutritional support that pure fasting cannot provide. Comment below and tell me whether you are going to try this protocol after today's lecture. And tell me what was the single most important thing you learned today. I genuinely want to hear from you because your feedback shapes the lectures I create. And now, if today's session gave you genuine value, hit subscribe and turn on the bell icon so you receive every lecture I deliver on this channel. Share this video with someone who needs to hear the real science behind what sardine fasting actually does inside the body. Thank you so much for your attention today. I will see you again very soon in my next lecture.